Good afternoon, friends. Welcome to the second edition of Elets Diagnostics Leadership Summit. We have with us in this session Dr. Debjani Saha, who is a Group Product Manager for Premas Life Sciences Private Limited, and she is here to give an industry presentation on the next gen sequencing, current and future paradigms for infectious disease diagnosis. I would now like to invite Dr. Debjani to give her presentation. Over to you, Dr. Debjani.
Thanks so much, and I thank the organizers for giving me an opportunity of speaking in this forum. I'm Debjani. I belong to Premas Life Sciences Private Limited. Premas, as a company, has been around for over 15 years now. We started off as a contract research organization, and we also have a very thriving life sciences department, and we bring cutting-edge genomics technologies to India, prominent among which is Illumina next generation sequencing. And my today's presentation would basically, chronicle some of the key applications of this, revolutionizing technology in the field of infectious diseases and microbiology. I think in the post-COVID era, the need for newer tools, cutting-edge applications for figuring out, the disease diagnosis and also to getting to the bottom of the pathogen has been felt like never before. And hence, it's very important that we understand how we can actually leverage these cutting-edge tools towards addressing some of these challenges which plague our country in terms of infectious diseases. So, in terms of the core methods in microbiology, we have these four sections, which pertain to next generation sequencing, among which you have whole genome sequencing, shotgun metagenomics, 16S rRNA sequencing, and virology, right? And I'll delve to each of these in a little bit of a detail, right? Now, in terms of microbiology, when we actually figure out a culture-based method, which has been, the method of choice since many years, if you compare that versus NGS, we have a lot of pros working for NGS, right? In terms of the amount of time that it takes to resolve a pathogen, so approximately a couple of days versus a week in culture-based methods. It's completely hypothesis free, does not require any a priori information. It's very, very sensitive, and it is scalable. You can actually sample thousands and several thousands of pathogens in a week's time, right? So, whereas culture-based methods are extremely low sample throughput, and of course, it generates such an important a volume of data, which has both potential for current and future analysis, right? In terms of whole genome sequencing, what are the questions that it answers, right? The few questions that it answers are: A, what is the pathogen? Has it been seen before? How can we fight it? Is it actually an outbreak, or is it just a one-off case, right? And if you just see, typically, if you just talk about culture-based methods, they take days to weeks in figuring out the pathogen, and often do not give important information about drug sensitivity, right? And we'll again see how, because some people might just say that culture tests will be able to throw some light on drug sensitivity, but then often that's not possible in detail, right? So the important thing is, when you actually talk about whole genome sequencing, that entire weeks to find out what your pathogen is shortened to 1 or 2 days, wherein not just would you actually figure out what is the pathogen of interest, but it also figures out a familiar lineage, right? You will be able to figure out what is the epidemiological lineage of that particular pathogen, and frame out a mediation strategy in place. Not just that, you will also get important information about the virulence or the resistance of the strain, and basically the drug sensitivity of the particular strain, right? And this is possible through the volume of sequencers that we have today. And not just that, you will also be able to correlate the data with a national databases, and also internationally when you actually share this data with collaborators. Especially because, and this kind of information is very important, especially because over generations, and we've seen that with the COVID strain, it's very fast in viruses, because viruses are naturally fast mutating organisms, but it's also there in a lot of bacteria, where generations after generations, when they actually replicate, they gather mutations, and some of these mutations make them very, very resistant to drugs, right? So, and that is our next challenge, really, because if we talk about some of the modern challenges of the healthcare system, it is really the antimicrobial resistance, because really we're running out of drugs to which our pathogens are sensitive, right? Again, um, NGS helps us, basically deconvolute a lot of important information, and we'll see what it is. So if you just see this particular example where you have these various strains from different sources and their drug sensitivity tests, we can figure out that all these pathogens are resistant to these two particular these antibiotics, right? But in these culture-based tests, they are not able to tell us what is the actual mechanism of the drug resistance. And you can see that through whole genome sequencing, we can get exactly that information. We're able to figure out what are the mutations in the various genes which render these pathogens, resistant to various drugs, and figure out a way on mediating this kind of a drug resistance, right? For the interest of time, I'll just move on to the next important application of microbiology, and that is metagenomics. And we again have a, you know, a, a what do you say, a short form of metagenomics and a long form of metagenomics, for the lack of a better word. Now, why is metagenomics important? Because the microbia in our body is very, very diverse, and it has a huge amount of role to play in the various things that we, exhibit, whether it's our, sensitivity to drugs, whether it's our, it's the effectivity of the nutrition we get, whether it's our disease predisposition, or whether we are able to basically be hale and hearty, basically for general wellness, right? So the microbiome in the human is very, very important, and it's basically the extra genetic quotient which contributes hugely to the well-being of a person, right? And needless to say, in the various ecosystems present in our body, whether it's oral microbiome, or the gut, or the skin, or the placenta, or the vagina, we have a rich diversity of microbia existing, and any change in this diversity can potentially be detrimental to the well-being of the individual, right? Now, how do we basically find out what this microbia consists, and why is it even important? Like I said, through various diseases or through various conditions, the percentage of the various microbia in our body get disturbed, and to find out what is wrong in any of these you know constitutions, it's important for us to be able to figure out what is the basal constitution, and then figuring out what has gone wrong, right? And that is why we have these two diverse ways of figuring out what the microbiome is. One is the 16S rRNA sequencing, one is the shortcut metagenomics. Like I said, 16S is rather a shorter form of the metagenomics and deconvolution. And the way you do it is basically profile the 16S region of the various bacteria. As we know that the 16S region is made of conserved and variable regions. So if you map out the conserved regions, if you map out the variable regions with having primers in the conserved regions, you will be able to profile the various 16S, constitution in your body, and basically get a sense of the bacteria which are prevalent. Shotgun metagenomics, however, it is a little more detailed, because often it basically allows you to find out. It's basically like hunting a needle in a haystack, because even if you don't even if you it's basically involved in profiling the entire genomic quotient of the microbiota, right? Not just would you get information about the various kinds of bacteria, virus, fungi, etc. present in the microbial population, but also you'll also get information about the genes which are present in the microbial population, right? And that is, like I said, that is possible through all of our benchtop platforms. And you also have a lot of these readily available pipelines which will give you very simple reports, right? So like I said, these are shotgun metagenomics, which is basically not just figuring out what is there in your microbial population, but also gene content, metabolic pathways, and total abundance of the entire microbiome taxon, right? And you have these various application notes which we can easily circulate to any of you who are interested in doing either the detailed microbial analysis or the 16S microbial analysis, right? Not just that, we're also able to do fungal metagenomics analysis through a very simple protocol, which is the ITS metagenomic sequencing. And we can, circulate the references to whoever is interested. And not just that, for shotgun, we also have really intense application notes. And this shotgun metagenomics, in fact, was seminal in finding out the SARS-CoV-2 when the infections first were seen, and people didn't know what was the pathogen which was really causing it. What was done was the RNAs were extracted from the nasopharyngeal swabs of various patients, and an analysis was done based on which they could actually figure out it was indeed a family of coronaviruses, and more specifically the SARS-CoV-2 virus. So any kind of a sequencing would basically, form these steps. One is the extraction. Extraction could be manual or automated. Next is library preparation, where you actually, bring the DNA or RNA converted into DNA to a form which is amenable to sequencing. Now, after that, sequencing happens, and then we have a lot of these pipelines available which can help you to analyze the data and get the data in a form which will help you not just to get the composition, but also more specific details on the genus and species information, right? And this is a typical class. This is a typical, report which is generated out of a 16S profiling, right? You get this sunburst plot, which will basically give you a classification of all the bacteria which are present, and also what is the relative abundance of the bacteria. Finally, as I know I'm running out of time, I just have a couple of minutes. So basically, I'll be delving on a little bit on the flavor of the season, which is basically COVID, and how sequencing has really helped us to figure out what are the mutations in the SARS-CoV-2 strains which are circulating in the population, and how you can actually, use it for disease surveillance. So key features. So basically, Illumina, the company, the genomics company, which is based out of San Diego, has brought out a test, last year sometime, which was initially it was introduced to do a high, like to do a high throughput detection of the SARS-CoV-2, but eventually it is being now used for disease surveillance. Now, basically, it's next generation sequencing based, and it detects the SARS-CoV-2 in the nasopharyngeal, oropharyngeal, and nasal swabs from patients. And it's highly accurate because it uses 98 amplicons across the SARS-CoV-2 genome to map the various mutations which are present in the various strains affecting the population. Uh, it is available in very, very flexible kits. So you have 96 sample kit and a 3000 sample kit available. It contains about 11 internal controls. So basically, when you do the reaction, you should be able to make sure that the human RNA is also captured, because you want to make sure that your samples are okay. So you have about 11 human gene targets in every reaction. And also, we provide a very, very simple pipeline which helps you to do the mutational analysis and generate a report, right? So, this is the basic test overview. You have your sample collection, you have your RNA extraction, you have your sample preparation, which basically involves the conversion of the RNA into cDNA, and essentially then you convert it into a form which is basically taken for sequencing, and then finally you have the analysis and the report generated. So essentially, like I said, once you have the samples, you also have an analysis option that we provide both on cloud and on a physical server. And this basically helps you in generating a report which is very, very lucid. And as of today, I think we've easily done about 60 to 70,000 samples on this kit across all the INSACOG centers, for mapping out the mutations in the SARS-CoV-2 strain and helping the government basically plan ways for disease surveillance. Thank you very much. This was essentially what I wanted to cover. I hope I'm on time, and I would love to take any questions that people have. Thanks.